Hi, in this video, I'm going to describe the mechanism of action of p53, and p53 is actually a tumor suppressor protein, and it's also help in cell cycle regulation, and also acts important player in the acts as an important player in the cell cycle checkpoint. So we will try to discuss one checkpoint and try to elaborate the function of p53 and how it functions as a key player of the DNA damage checkpoint. So here we have a cell, a healthy cell. So inside the cell we have the nucleus, and so here we have our DNA inside the nucleus, and due to some reason there is a ionizing radiation, and this ionizing radiation causes a damage in this DNA, and as a result there is a double strand DNA break, which is actually very uh, common DNA lesion. And now we would see what happens and how cell behaves when there is such kind of DNA lesions. So if we just zoom into this portion, so we would see that there is a DNA lesion, double strand DNA break. In this situation, if cell allows it to divide, then it would be quite dangerous, because in the daughter cell these there would be a uneven segregation of the genetic material, so unequal distribution of the genetic material, so that is not expected. So that is why cell have to devise a mechanism by which it can actually tell the cell to repair this kind of damage, at least try to repair this kind of damage. And so this is actually regulated by p53. So before knowing p53, so we have to understand that there are some factors which actually helps in the recognition of these lesion, DNA lesion. So such as for instance, say this is a UV ray. UV ray actually damage the DNA. So there must be some sensors which actually sense this kind of damage. So one type of sensor is ATR, or another type is ATM. So ATM stands for ataxia telangiectasia mutated in yeast. So whatever, we should think it as a sensor. It would sense that there is a double strand DNA break, and it would tell its friend, which is actually a kinase called check1 kinase, and this check1 kinase would perform a particular function. So this check1 kinase would phosphorylate a downstream protein, and let's see what is that particular downstream protein. So in the meanwhile, in the cell, what we have in p53 is actually inhibited by another protein named as MDM2. So MDM2 is actually inhibiting p53, but p53 gets phosphorylated by check1 when ATM senses are ATM or ATR senses are DNA damage. So then p53 is actually phosphorylated. So then MDM2 can't hold p53 any longer. Now p53 is now free, and p53 has multiple transcription targets. So one of the transcription target of p53 is p21. So as I have discussed earlier in my videos, that p21 is actually a inhibitor of S-phase cyclins. So S-phase cyclins are, like, if we name them, then cyclin it is cyclin E and CDK2. So what happens that these p21 actually inhibits these cyclin E and CDK2, and cyclin E CDK2 can't perform its function. It can perform its function. As a result, what happens? So as a result of these inhibition of cyclin E and CDK2, what would happen? Cell cycle progression stops, and now cell would get some time to actually divide. Cell would get actually some time to think, and it would try to repair the DNA. And if the DNA is repaired successfully, then cell would resume its division. If the repair is not successful, or the damage what is caused by this UV radiation or any kind of radiation is very harsh and very serious, and it is irreparable, then what happens? The cell would undergo programmed cell death, also known as apoptosis. So in this way, p53 along with p21, which is a cyclin dependent kinase inhibitor, helps to regulate the cell cycle when there is a DNA damage. And this is the basic molecular mechanism of DNA damage checkpoints. Hope you enjoyed the video. Please like and subscribe. Thank you.